This is preparation recording for lecture 3. We are going to talk about the basics of DNA structure and replication. This course is not primarily molecular biology, but genetics. So understanding the structure of DNA, what nucleotide sequences are, and how changing the nucleotide sequence changes the protein. That's foundational to much of the other stuff we're going to be talking about, but the focus isn't necessarily just memorizing every enzyme or anything like that. So take your cue from the level at which we're talking about it. You will be taking molecular biology courses that will go into more detail. Okay, and so I've set a few learning outcomes. I may add to them before I post the notes, so just take a look at them. The learning outcomes that I include are the things we hope you will know and be able to do once you have been through all the prep and all the practice for a particular topic. So it doesn't necessarily mean that just listening to a lecture is going to be enough. Sometimes you may have to practice. And we're going to provide lots of opportunity to practice both in the tutorials and on the Codon website. Okay, so for this topic, my plan is for you to get the basics of the experiments and the essentials of DNA structure in advance. So I'm going to go over that here in this recording, and there will be other resources posted. Maybe some more interesting videos and things like that for you that are available. And then in the lecture, we'll be touching on those key points, the basics of the DNA structure, especially things like the polarity and the complementary base pairing. Those things are really important, essential for DNA structure. And then we're going to go through a kind of tutorial / quiz. I will just take you through all the steps of DNA replication at the level needed for this course in clicker questions in the lecture. And so just a warning, this will probably be divided into 2 recordings, and they're not going to be 5 minutes. They're going to be more like 15 or so, maybe more minutes each. So if you already know a lot, or you've just recently finished molecular courses, or you just happen to have some background or plan to read up on it yourself in one of the textbooks that I'll make available through the library, you might not need this. So you can skim through the notes, and if it all looks pretty familiar and reasonable to you, you don't have to listen to this. It's only if you feel that this will help you that you should go ahead and spend the time. Okay, so let's just start. So it was in the sort of mid-60s when the Watson and Crick structure of DNA was first published, and so it's around a little more than 60 years ago. And so this is what we knew, and we're going to go through some of the evidence for some of this, as well as kind of the DNA structure. So it was known that genes, what were called genes, units of heredity, were associated with certain character traits. The fur color of mice, and whether they had long or short tails, and flower petal colors and all that sort of thing. And it was known that there were mutations, changes in genes that altered gene function. So mice that had albino fur, all white, instead of having the normal brown kind of speckly color fur. So it was known that gene function was altered by mutation, but it wasn't completely clear what the nature of that was. It was known that a gene was equivalent to a polypeptide. This is a little oversimplified based on what we know, but the idea was that genes were somehow associated with a protein and a functioning protein. Genes were known to be found on chromosomes, and chromosomes consist of DNA and protein. And for some time, it wasn't clear whether the DNA was the genetic material or the protein. And DNA is not very variable. There's only 4 different nucleotides, whereas with proteins you had over 20 different amino acids if you include some of the modified ones. And so protein seemed like a better choice because there was more variability. However, by the early 60s, we did know that DNA is the genetic material. And so we're going to go through some experiments that demonstrated that, and then a little bit more on the experiments that told us about how, what the structure of DNA is, and then we'll go through replication, probably not until the next recording. So when I go through these experiments, I'm going to tell you how they were done, and I'm going to tell you the results, and then I would like you, before class, to think about this question. So just think about the experiment and show and how they actually showed that DNA rather than protein was the genetic material. That's what the next two experiments with bacterial strains and then with viral strains as well showed us. Okay, so the first experiment we'll talk about is the experiment done by Griffith, and Griffith and the people in his lab, of course, worked with two different bacterial strains, and they had two strains. One was just called the R strain and one was called the S. They were the same species, but they were variant types of that bacteria, and one of them was considered non-virulent. I'm just going to put non-V. Or no, I'll write the whole word so that you can see. Virulent. And that meant that you could expose the mice in your research with this bacterium, and they survived just fine. They were not made sick, and they did not die. But there was a variant strain that had just a little bit of difference. And if you infected the mice with this S strain, they would get sick and most of them would die. So that's what virulence refers to when we're talking about a bacterium, the ability to cause illness in the people, the animals, the organisms that are infected with it. But it turned out if you heat killed the bacterial strain S, so you boil it, you kill all the cells, and they break apart. And if you infect mice with the broken up material, they also will survive, right? So by heat killing the S strain bacteria, you created a compound, a mix of materials that was not virulent anymore. And so the key experiment was if they took live R strain, remember those are the non-virulent ones, and then the material that came from the bacteria that had been heat killed, either by autoclaving or boiling, whichever method they used. And when you mix these two together and then expose the mice to it, excuse me, the mice would get sick and most of them die. And when you cultured tissues from the poor little mice that had been sick, you could isolate S strain cells. So, and of course they would repeat this to make sure that they hadn't just missed, killing all the cells. And so with the appropriate controls, it became apparent that somehow when you provide non-virulent cells, but then something in this mixture changed the R strain into the S strain and made it virulent and made it kill the mice. And so that was the first key experiment. And this was followed up by a group of people that asked the question, okay, what component in here is responsible for changing the R strain into the S strain? And they did a series of what you would call maybe elimination steps. So they would have a control. So they would take the S strain and boil or autoclave it so that all the cells are dead. And they would use that extract and they would expose it plus R strain to the mice, and the mice would die. And then you would take a subset, a little sample of each of these in a test tube. And each one of them was treated separately. So in the first one, they used enzymes that destroy polysaccharides, sugars and starches. And if you destroy those components of this extract, it doesn't alter the fact that the R strain gets transformed into the S strain, right? You can do the same with lipids. So you use enzymes that destroy lipids, the cell membranes and the different kinds of fat type molecules in there. And that doesn't work either. You can destroy RNA using RNase enzymes. These are ones that break RNA to pieces and dismantle it. That didn't fix the problem. You could also do the same with enzymes that destroy proteins, and that didn't do it. But if you destroyed the DNA with enzymes that specifically degrade just the DNA, you have now prevented, you have killed the component of this sample that conferred virulence on the R strain, changed the R strain into virulent, and these are happy mice because they have survived this. And you could culture their feces or do swabs of their bodies and so on, and there was no live S strain recovered. Whereas from all of these, when you cultured the tissues, they were infected with S strain bacteria. So when we talk about the transforming agent, we're talking about the component of this extract that changes, transforms the R strain into S strain. Okay, so that's the result of that first experiment. So, and both of these, so Griffiths and his lab did this work early on, sort of late 20s, I think. And then it was a while before this work was extended with the sort of elimination. If you eliminate different components individually, you can find the one component you can eliminate and eliminate the effect that you're studying. And then phages are a type of bacteria that look like spaceships to me, alien spaceships. They specifically infect bacteria. And so this experiment was done with two different samples. One sample was radioactively labeled with S35, radioactive sulfur, and that labeled protein. And then there was another batch that had been incubated with P32, which is radioactive phosphorus, and this primarily labels DNA. So what you have now is you have viruses that have radioactivity, both of them, but their components are differently labeled. And then you allow them to infect bacteria. This is the bacterial cell, and then this is the little phages. And phages infect bacteria, by introducing their genetic material, and then they basically take over the material, the enzymes and the machinery inside the bacterial cell, and they turn it into a cell that just produces more and more and more of all the components to make more viruses. So the cell becomes a virus factory, and eventually all the viruses get built, and the cell bursts, and the viruses spew out in all directions. That is how viruses infect these cells. And so the way this experiment was done is the infection process was allowed to proceed for a short period of time, and then you would put the cells and the phages into a blender. When you do this, it disrupts the process. So the phages are peeled off of the bacterial cells. And then you can centrifuge. So you put them in a tube and you spin. And the bacterial cells will be in one place, and the phage ghosts, they're called ghosts because they don't contain all the components anymore. And the phage ghosts will be found somewhere else. So this is where the cells will be. Cells. And this is where the ghosts will be. And then the idea is, where is the radioactivity? And so in this experiment, the radioactivity is all found in this part of the tube after centrifugation, where the lighter components are in the ghosts. Whereas in this experiment, you have your two different components, and the radioactivity was found in the bacteria. And the bacteria have taken in the genetic material to produce more phages, right? So based on this experiment, it was determined that the phage genetic material, the material they use to change the bacteria into a little factory for producing more phage, is the DNA component and not the protein component. Okay, so I want to go through some basics of the DNA structure, but I'll point out here that I am not expecting you necessarily to be able to draw these in detail. If you do, then that's fine. There's no problem with that. But what I would like you to be able to draw is the ribose sugar, which is a pentagon, to be able to put the base A, C, T, or G on the 1' carbon, to be able to identify the 3' hydroxyl group. That's what this is. And then the phosphate group on the 5'. And if you can draw this is enough of the structure for the purposes of this course. Knowing every single, nitrogen and oxygen and so on is not, it may be useful and necessary for other courses, but it won't be for this. But I am showing you this because the nucleotide, there are 4 different nucleotides in DNA. And the things to remember are what a purine is and what a pyrimidine is. Purines have 2 rings, and they are A and G. So adenine and guanine, and you might as well know the names too. And then the pyrimidines are the ones with single rings, and they are cytosine and thymine. The way I remember which is which is that I remember that purine is the shorter name, but the bigger molecule, because it has two rings instead of 1 ring. And those are the kinds of things I often use to help me remember or figure out how something works and so on. So I'll be passing them on to you. There's no need for you to use these unless they're helpful to you. But I do tend to share them just because I think it could potentially be helpful for some of the students. So these are this is a close-up of just sort of nitrogenous base adenine, A. Again, as I said, you don't need to remember this. If you could draw just an A here or write the name adenine, that's fine. But I'm pointing out that the carbons are, so this is a ribose 5-carbon sugar, and it has 5 carbon atoms in it, and so they're numbered 1, 2, 3, 4, and 5. So the things to pay attention to is that the base is added to which is this part, these rings. These are called nitrogenous bases because they have their rings with a lot of nitrogen in them, and they are attached to the 1' carbon. The 3' OH is very important because the OH group on this carbon, this is where new nucleotides are added when DNA is being synthesized, right? So this is a very important part. And then the phosphate, which is the other end of the molecule, is added to the 5' sugar. And when DNA is synthesized, the 5' phosphate of one molecule is added onto the 3' OH of another molecule. And so we always say the molecules grow 3' to 5' because they are, 5' to 3' because the new nucleotide keeps getting added on the 3' end, and so it extends sort of in the 3' direction. I also have an arrow here, and that is because the 2' carbon is the reason for the name deoxy. So deoxyribonucleic acid, deoxyadenosine 5', in this case monophosphate. The deoxy refers to the fact that it's because of comparison with RNA. RNA has an OH group here, but DNA does not have that, so deoxy is sort of saying it's missing that second oxygen. Let's see. I think that's everything that's important to know at this point. And then we'll finish with Chargaff's rule. So Chargaff was, and his research group were studying sort of the components of the different bases of DNAs from lots of different kinds of DNA. Does the DNA from a human and a sea urchin, for example, have the same amount of adenine, thymine, guanine, and cytosine in it? Or do they differ? And if they differ, what does that mean in terms of the different species, the different, features of the organism, their different physiology, and all this kind of thing. And so in a number of organisms, this isn't everything, but this is the stuff that was reported at the time. They found, for example, when looking at one strain of E. coli, that you had almost the same amounts of adenine, thymine, guanine, and cytosine. And so this was, this is of interest, but this is not very useful if you're trying to understand sort of how the molecules work. However, if you looked at a different species of bacteria, there was considerably more adenine and thymine and considerably less guanine and cytosine. So now we're seeing differences, and the differences are what might tell us something about how the molecule is structured. Okay. Here's one. This is another kind of bacterium, but in this case it's different. The adenine and thymine are much smaller, and the guanine and cytosine, oops, sorry about that. I am left-handed, but I have to use the mouse with my right hand, and so that means I'm not very good with it. Okay. So there's some things to notice here. One is that each different species has different approximate concentrations of the different nucleotides. And if you look at the ones where there is a difference, the adenine and thymine are similar to each other, and the guanine and cytosine are similar to each other. And noticeably so, 32 to 33%, 17 to 18%, right? And so this helped researchers determine something about what we call complementary base pairing. So before the next video, just give that some thought and then ask yourself, what would this suggest? If the amount of adenine and thymine were always pretty much the same, and then the amount of guanine and cytosine were always pretty much the same, what does that actually tell us about how the molecules are put together?